Hello to everyone. First of all, I would like to thank the organizers for inviting me to participate in this very interesting symposium. Today, I will discuss about the present and the future of human genome engineering. So first, let me introduce a bit myself. So I'm the principal investigator of the Translational Synthetic Biology group at Power University. And basically, our group is devoted in developing new biological technologies. So more or less 2/3 of the lab are devoted to creating new technologies for gene therapy. And the other third is developing technologies and modules for skin microbiome engineering. Today, I will not discuss about our research, so I mainly will give an overview of the field of gene editing, where we are now and where we are going. So, well, I'm sure you are very well aware of this massive revolution that biology has experienced. So first, a read revolution. So we have increased our capacity of sequencing massively. We can sequence a human genome today in a few hours. But actually, we have also experienced an incredible revolution on writing DNA. First, on synthesizing completely novel DNA. Maybe you have heard, right? We have already synthesized fully synthetic chromosomes of bacteria, of E. coli, for example, or mycoplasma, or even of eukaryotic organisms like yeast. And also, we have been capable, or we have really increased our capacity to edit DNA. And here is probably the main revolution that we will discuss today. So basically, essentially, this protein, this magic protein, which is Cas9, you see here how it recognizes the DNA and how that makes a cleavage where it finds a target sequence, which has really, I think, accelerated research and also translation into therapies. So this is just what I was mentioning. So I think it's very clear the impact of gene editing technologies in scientific research. These are papers citing CRISPR. You see this exponential growth, which probably is still continuing. And then this enhanced capacity to engineer genomes actually not only has accelerated basic research, but as we very well know, is also accelerating translation into very exciting therapies. This is just an overview of clinical trials, basically engineering humans for curing different types of diseases, cancer, genetic diseases, and others. And you can see that the numbers are quite big. So genome engineering has definitely accelerated discovery and also translation into new therapies. This is the outline for today. So I will spend most of the talk discussing about the present. What can we do today? Which actually is quite a lot. And then I will, in the end of the talk, I will focus a bit of the future and from a technological point of view. So I will not get too much into any ethics. Definitely, I think there are speakers way better prepared than me to discuss about the ethical and social implications of gene editing. Okay, present. Basically, I'll just very quickly introduce CRISPR. I'm sure that many of you you know what it is, but just a brief reminder. So actually, CRISPR is an immune system of bacteria, which basically it's a capacity that the bacteria have to attack invading DNA. So somehow they can build some memory, what is called this CRISPR array, memory of, for example, phages that infect this bacteria. And then there is an army of genes that can use these small RNAs to identify as soon one of these viruses enters into the cell and really cleave the DNA or the RNA and trigger this interference. Actually, some of these Cas genes is what has been really revolutionary for mammalian genome engineering, for example. So actually, there are many types of CRISPRs. The ones that we usually use for mammalian applications, so for example, for gene therapy or engineering of embryos, actually is some compact CRISPR types, which basically the recognition of the target DNA and the cleavage is done by a single protein. Probably the most famous of all is Cas9. It's a quite large protein that does all this process. Maybe you have heard also about Cas12, which is another relatively famous CRISPR protein. And finally, you may also have heard about Cas13. Cas13 actually recognizes RNA instead of DNA. But how do we use these proteins? Like, for example, Cas9 to engineer genomes. So how do we use CRISPR to engineer mammalian genomes? So actually, we have developed different methods and that exploit different mechanisms. So probably the most used is using DNA cleavage. So the classical biological activity of CRISPR, which is recognizing DNA and doing cleavage. And here, basically, we then exploit the natural repair machinery of the cell to induce different genetic changes. However, scientists have brought CRISPR well beyond that. And actually, we'll see methods where we don't need to rely so much on the DNA repair, and actually we can directly replace bases. So we can, change bases, what is called the base editors, or even replace bases, what is called the prime editing. So we'll see these methods a little bit more in detail. So first, let's start with the cutting. So how do we induce changes in genomes by this programmable nuclease activity? So actually, we mainly exploit two pathways. After double stranded break, we can either induce de novo joining, and this is very much used to break genes, to do knockouts, and you will see that this has been quite successful until now. So basically, we use CRISPR to target a certain position in the genome, induce double stranded break, and then the same system basically will accumulate different deletions, insertions, which will cause loss of frame. Or on the other hand, we can supply a donor template together with the CRISPR system to induce a mobile recombination, and then we can, in this case, program specific changes to the cell. So let's see different CRISPR systems or different CRISPR protocols that we use to do different types of gene editing. We will start with the simplest: how we can use CRISPR for inactivation of genes. In this case, we will exploit the left pathway, the non-homologous end joining. And actually, what do we need for that? Actually, we need only two components. We need the Cas9 protein, which will do the recognition and cleavage, and we'll need the gRNA, which will program where this Cas9 needs to go and do the cleavage. And this is how it goes. So we combine these two elements, we put them in the cell, and basically the cleavage will be made. And actually, Cas9 will keep cutting if this relegates without changing the sequence, and actually will keep cutting until the repair machinery makes mistakes. And that's why this is so effective to do, for example, knockouts, because we tend to accumulate insertions, deletions, or sometimes mutations. It may sound counterintuitive, but actually doing deletion of genes is quite useful, even in therapy. Actually, this was a review from April last year. And actually, you see how many clinical programs, actually, they exploit CRISPR to do knockout. For example, knockout of immune-related genes, in, for example, immunotherapy, or we'll see even one application in sickle cell anemia. Or here, for example, here we see how we use CRISPR to delete one exon, which actually is causing the loss of frame in this case, in Duchenne muscular dystrophy. And actually, in this case, we use CRISPR to break, to somehow force exon skipping. So in this case, and this actually is a program brought to the clinic by Exonics, recently acquired by Vertex. Another example where breaking things can be helpful for therapy, in this case, is an example of lobar congenital amaurosis, which is a genetic blindness. And in this case, basically, actually, it's quite challenging because the gene that is affected is very large and it doesn't fit into an AAV. And here, the company Editas Medicine, actually, they developed a method where they can edit in situ the cells. And how do they do it? Actually, the mutation that they have addressed causes a cryptic intron to appear and make the protein lose the correct reading frame. So then they use CRISPR actually to excise, to break this cryptic intron so that they can reconstitute the frame of the protein. And actually this is the first in vivo CRISPR therapy that has gone to the clinic. Then also another exciting field for CRISPR applications is blood diseases, in this case, sickle cell anemia, beta thalassemia. And actually one of the most advanced programs actually is in using CRISPR to break a regulator which keeps dormant our fetal hemoglobin. So in this program, the company CRISPR Therapeutics, what they are doing is an ex vivo therapy where they inactivate this regulator in hematopoietic stem cells so that basically they can wake up this fetal hemoglobin. Actually, this was the first. So actually, you can see the shares of these companies sponsoring this trial, CRISPR Therapeutics and Vertex. And actually, you see the big spike. And actually, this was caused by the first data we got on effectivity of a clinical trial using CRISPR. In this case, they observed in 2 patients actually positive outcome of this CRISPR therapy for these hemoglobinopathies. Another exciting case that was published, I think was last December, last January, if I don't remember back, and actually is using CRISPR in this case to engineer T cells to improve cancer therapy. Again, an ex vivo therapy where the T cells of the patient are extracted and then modified with CRISPR. In this case, they use CRISPR to first accelerate how these T cells can attack the cancer by knocking out the PD-1, well-known break for the immune system, and also making a space for a tumor-specific TCR receptor. So they knock out the endogenous TCR receptor, and then they use a lentivirus to put another TCR receptor that will basically recognize the cancer. Actually, this was a phase 1, so they evaluated safety. It seems that this treatment was proven safe, but more research will be needed to prove the efficacy. Okay, so we have seen how we can delete genes with CRISPR and actually how this can be very useful in therapy. We saw already 2 examples in the clinic. So can we use CRISPR also to construct new alleles, to do homolog recombination, in this case, exploiting the other pathway where we put a template and we really want the DNA to acquire this sequence, which we program by this donor template. How do we do that? So basically in this case, we need to add a 3rd element. So of course we need again Cas9 and gRNA to program this specific cut. But in this case, we'll need to supply a 3rd thing, which is a template, which will direct the repair of this DNA sequence. I have to say that this is normally less efficient. And actually, you may be aware that homolog recombination is only effective in cells that divide. So in this case, we have a tissue limitation. We can do hematopoietic system, we can do maybe T cells, but actually most of the tissues of the body, this will not work. And here again, we have an example on basically editing the beta globin gene, in this case directly. So we saw a solution by breaking the regulator that keeps dormant the fetal hemoglobin. Here we'll see directly editing the mutated sickle gene. And in this case, it's worked by Matt Portheus. And in this case, they directly use homolog recombination in hematopoietic stem cells to really repair the mutation that is causing, in this case, the sickle cell anemia. And actually, this project actually was recently announced that it has been funded by Bersen to really bring this to the clinic. Okay, what other things can we do with CRISPR? So we have seen how we can break genes, how we can repair genes, but actually another very exciting application is to insert new genes. And here again, we use the repair of the repair machinery of the cell. So different methods have been proposed using homolog recombination again, and also non-homologous end joining, and also microhomology related end joining. So let's see probably the most advanced one, which is exploiting non-homologous end joining. What do we do in this case? In this case, basically, what is this method called HITI? You may have heard. It's actually quite used in the field. And basically, here what the researchers propose is to simultaneously cut the target insertion site, so where in the genome you want to insert the gene, as well as the donor DNA. And then somehow they induce this non-homologous end joining mediated insertion of the gene. Of course, this has the advantage that it can work in most of the cells, because most of the cells will do non-homologous end joining, and actually it's quite used in the field, this method. And actually, one very interesting applications is for hemophilia. So basically, the company Intilia is using a very similar method to basically deliver Cas9 gRNA plus an AAV containing the factor 9 to the liver, and basically doing targeted insertion of factor 9 using this similar method that exploits non-homologous end joining and Cas9 to insert a correct version of factor 9 into the liver. Also, it has been proposed to use homolog recombination, of course, in the tissues that homolog recombination works. And here again, we go to the case of sickle cell anemia. So we can also basically not only correct the mutation, but also insert a full new gene directly in the site that we would like to. In this case, of course, limited to cells that divide like hematopoietic stem cells. Okay, so we have seen a bit how we can do many things using cleavage, using Cas9 to cut the genome and then induce repair in different directions. But actually, the field of CRISPR has invented completely different solutions to edit genomes. And actually, a very exciting example is base editors. Here, we are using Cas9 to direct some protein machinery somewhere in the genome to basically directly modify the DNA base. Basically, these are how they look, these elements. Actually, we use a Cas9 that is partially inactivated, so it will not do a double-stranded break. And then we use a protein, in this case, a deaminase, which will transform, for example, a C to a T. So basically, it will deaminate a cytidine to create a uridine, which will be read as a T. Or more recently, they have also developed, this was work done in the Liu lab at Harvard, they have developed an adenosine deaminase, which will deaminate adenosine, and it will be basically converted to inosine, which will be read as a G eventually. So here, we are not using a double-stranded break to induce repair. Actually, what we are doing is directly change the bases. That's why the name of base editors. We can change one pyrimidine to another pyrimidine, or we can change one purine to another purine. So very exciting. So DNA double-stranded break-free genome editing. A bit more details on how these systems work. Actually, they became quite sophisticated. So people have done several iterations on improving them. So for example, in the case of cytidine base editors, to change C to T, actually they also incorporate extra functionality. For example, of course, if we put a U, so the first step is basically modifying the C to become a U. But of course, the DNA machinery of the cell will recognize this U. So then this system, it also incorporates uracil DNA glycosylase inhibitor. So then basically we protect this U in the genome. And then in addition to that, of course, here we have a mismatch. How do we tell the system if it should repair the upper strand or the lower strand? Then what we do is we use a Cas9 that will cleave only in one side. And then we'll tell basically the DNA machinery, please repair this strand, not this one. So then we can basically establish our desired outcome, which is changing this GC pair to a TA pair. So quite exciting work. And then also something similar has been developed for adenosine to transform it into adenosine. Okay, so we have seen how we can use double-stranded breaks. We have seen how we can directly change bases. Now we'll see how we can directly replace bases. This is something quite new. It was developed less than a year ago by the same lab, actually, David Liu at Harvard University. And in this case, what they do is directly use Cas9 to search for the sequence that you want to replace. And then in this case, they attach a reverse transcriptase to the system that will in situ synthesize some new DNA that will have programming. So in this case, we'll use a more complicated gRNA. As you see that the gRNA is quite long. So one part will be the classical gRNA that will program Cas9 to go to a certain place. But then the other half of the gRNA will encode the piece that you want to replace of DNA. So in this case, for example, if you want to replace, let's say 10 bases, you would encode this new 10 bases here. So Cas9 would bring to the place of replacement, and then the reverse transcriptase will synthesize the new DNA. This has been demonstrated recently, so very exciting prospect is it allows to do insertions, deletions, substitutions, short insertions, deletions, substitutions, and it seems to be pretty much DNA repair, not very dependent to the DNA repair, so very likely works in many cell types. So it's, I think many people have lots of hopes with this new methodology. It has some complexities because the design is a bit more complex than, for example, in the case of classical Cas9, but definitely a very, very exciting work. Okay, so we have seen a bit the toolbox, the CRISPR toolbox. So what we can do with CRISPR. Let's see in which ways we can use it. So basically we can use DNA ex vivo, right? Take out the cells from the body, modify them and put them back. We have seen that. We can also do it in vivo, so directly modify in the body, or we can also, we'll discuss a bit how we would do germline editing and what has been done in germline editing. So first, ex vivo, we already have seen a couple of problems of ex vivo. So in ex vivo is where we have the luxury to take the cells out from the patient, modify them and put them back, which is technically sometimes easier than doing it in vivo. So in this case, probably the most famous case is CAR-T therapies, where we can genetically program the T cells to attack the cancer. So in this case, we remove the T lymphocytes from the patient, and basically we modify them to add the CAR gene, which basically will guide the lymphocytes to the tumor, and then will activate them to attack the tumor. And we have a couple of drugs approved. I think it has been very, very exciting prospects. And also we have seen what I mentioned before, ex vivo therapy for sickle cell anemia, beta thalassemia, and also ex vivo therapy for another immuno-oncology application in this case, which was basically adding another T cell receptor that will attack directly the cancer-specific T cell receptor, in addition of accelerating the aggressivity of these T cells to the cancer by eliminating the PD-1. In vivo, we have seen many examples already of gene therapy in vivo as well. Pre-CRISPR times, we had already some gene therapies, for example, LPL, which was approved quite a long time ago. Basically, it's an AAV-based therapy where basically the therapeutic gene it's put inside of this vector, and this is for LPL deficiency. Also, more recently, we have seen medicines developed for, again, another type of lower congenital amaurosis, for a type of blindness. Basically, in this case, the therapeutic gene is again put into an AAV and injected in the subepithelium of the retina. Or also very recently, you may also have heard about this AAV therapy for spinal muscular atrophy. Also very exciting therapy by Novartis. And all this pre-CRISPR, so no CRISPR involved. Basically a viral vector, another AAV vector that encodes a therapeutic gene and delivered to the corresponding tissue. Here we mentioned before that also CRISPR has been used in vivo, at least now in clinical phase, not approved yet. Clinical trial phase, I mean. We saw this before. And what about germline? I would really like to discuss a bit the technical aspects of germline in these last minutes that I have left. Of course, modifying the germline is a bit different from what we have seen so far. So far, we have been seeing ways of modifying the somatic cells. So germline means basically modifying some cells that could perpetuate along the time. So basically in the subsequent generations. Very quickly after the CRISPR methods development, there was, I think, pretty much a consensus on a moratorium, so that these techniques shouldn't be used in human genome germline engineering. While there is a big consensus that the somatic modification of the human genome is quite beneficial, and actually we have seen that this has already been demonstrated. So for germline, there has been this, I think, wide consensus that we need to be very careful. A brief history from a technical point of view. So actually in 2015, I think we saw the first report of doing CRISPR on embryos, in this case on non-viable embryos. This work carried out in China. In 2016, we saw basically some basic science work on human embryos in modifying the OCT4 gene. Some work that has been followed up, and actually it has explained a lot about the science of engineering human embryos. Actually, in 2017, this work by Mitalipov, basically on editing, on correcting, in this case, using CRISPR to correct the mutation on a human embryo. In this case, there has been some controversy on the mechanism, because in this case, the paper was pointing to some gene conversion. Some people has argued that maybe the mechanism was towards another direction. So we'll discuss a bit about that. But anyway, very exciting pioneering work. And then I'm sure you all have heard about the Güell in China, that he basically genetically engineered embryos, especially famous the case of Güell and Nana, that basically the scientists tried to inactivate the CCR5 to achieve HIV resistance. So actually, he basically used CRISPR and so Cas9 and the gRNA to inactivate the CCR5 receptor. Basically, it was not achieved completely. In some cases, it was. There were also levels of chimerism, it seems, even though we don't have all the information of this experiment, of course, it has not been published in a scientific journal. And yeah, it triggered a big international outcry, and he actually faced legal charges in his country. Actually, how he did it, well, you see, basically he directly injected Cas9 and the gRNA targeting the CCR5 loci into the embryos of the single cell embryos of these babies. The embryos were grown a bit, implanted, and then basically the babies were born. But the first question that maybe comes to our mind, to many of us, is it safe to do this? Here again, I will not enter into ethical issues because I think there is people way better prepared in this symposium to talk about this. Actually, we got very interesting work published this June, so far in bioRxiv, but from very good labs that we can analyze a bit how safe is this procedure. And actually, it's quite surprising that actually doing a similar process in another gene, of using Cas9 and gRNA to inactivate the gene, actually we observed big amount of genomic vandalism. What do I mean by genomic vandalism? Actually, big losses of DNA material, and even losses of big DNA rearrangements, and even losses of chromosomes, and actually in a quite significant percentage. So that's definitely at least a red flag that we need to see what we can do about it. Another work actually published the same month. Actually, they saw some things on the same direction. So they did basically also CRISPR on the embryos, and actually there was also again abundant chromosomal loss, and actually very important also a quite significant amount of mosaicism, which creates very difficult to guarantee the outcome of this gene editing process. Okay, so this is what I had for what is the present. So let me use really the last 3 minutes to discuss about the future, to see how we can address some of these problems, and some that maybe we don't have the solution yet. So first, well, I think it's clear that at least regarding this China case, as my actually previous mentor was saying, right, the genie is already out of the bottle. So I think it's very good to accelerate the debate of how we should really make the best use of these technologies on a more reasonable way to help patients and help humanity in general. First, from a technical point of view, can it be made safe? Can we make disappear these issues of chromosomal abnormalities, genomic vandalism, or mosaicism? Actually, for the on-target vandalism, I think that new CRISPR technologies like base editors and prime editors, since they don't do the double-stranded break, I think they should reduce significantly this process. Of course, we'll need to demonstrate that. We need to do some basic science to really demonstrate that, but that's my hypothesis. Regarding off-target has been largely discussed in the context of gene therapy and in the case of course, germline editing. And actually, I think in most of these studies, they observed off-target, definitely in the case of G&K and also in some of the work published last June. So these, I think we have also good solutions. Of course, we'll need to demonstrate them using the adequate basic science, but the modern systems, they greatly reduce the off-target, almost to undetectable levels. However, my main issue still is basically regarding efficiency. I think today we don't have any guarantee that we have a protocol that tomorrow will give us enough efficiency to guarantee absence of mosaicism. Of course, I'm sure these are things that we can improve, and more basic science will help in seeing whether we can correct all these issues that these methods have today. Happy to discuss more about this on the questions. I didn't want to extend myself too much. And then from a gene editing design, this is from the execution point of view. Of course, you know, biology is complicated, epistasis, moonlighting, individual variation, all these are major effects that will need to be analyzed case by case. So we know, right, some people have experimented in, for example, this is not therapy, this is more like disease resistance or enhanced properties. People have experience with some alleles with large impacts. This is a list made by George Hart. And basically, we know that some people have tried them in a bit unconventional context, like this biohacker that was trying to inject the CRISPR cocktail to break myostatin. Of course, was not very successful from what we know. Then we had, like, of course, what we just mentioned of the Chinese scientist that he tried to inactivate CCR5. And actually, CCR5 is an interesting example to discuss. CCR5 basically, of course, gives some resistance to HIV, but also it does other things. For example, makes us more sensitive to the flu, makes us more sensitive to the West Nile virus. And actually, in mice, it enhances cognition. So definitely biology is complicated. And besides the technical aspect of how we will implement this change, there is also lots of issues to discuss regarding basically the complexity of biology by itself. And then finally, you may also have heard, this is not germline editing, but this company BioViva that is commercializing anti-aging therapies. Basically, the CEO of this company injected herself several years ago with extra copies of telomerase. Again, something that should protect from aging, but of course, there are some concerns about this. So yeah, this was like just to put a bit the functional aspect, which is also very important to analyze. And yeah, and to sum up, just ready to close here, I think somatic gene editing seems to be safe. We have seen some clinical problems, at least with, we see some degrees of being safe. And even we saw some efficacy to treat serious medical conditions. And I think there is a growing number of promising clinical programs. So I think this is a field that clearly is growing and actually will give for sure a good benefit to humanity. Regarding germline editing, well, we see that the technical foundations are relatively mature. So far, the main problems are mosaicism and also genomic vandalism. And these processes are not fully well understood. For some, we may have solutions. For others, maybe too, but maybe a bit with a bit more work. And yeah, and with that, I would like to close, and I'm very happy to take questions.